welcome everyone, and welcome back to many of you. But I'd like to also welcome those who are now joining us from other research groups and institutions. So today we're going to hear from Associate Professor Sarah Bauermeister, who is a cognitive neuropsychologist and epidemiologist, and she works in the Department of Psychiatry at the University of Oxford in the UK. She is the senior scientist for Dementia's Platform UK, where she's been working for, I think, 7 years, she said. And DPUK is a cohort data repository for over 60 cohorts and holds data for over 3.5 million participants. From our end, we're very grateful for the support that DPUK has lent to the development of Dementia's Platform Australia, which is led by the Centre for Healthy Brain Aging, and for their collaboration on our COSMIC consortia. Now, as well as being an academic researcher with her own research project, Sarah leads the programs for DPU data curation, which we will hear a lot more about in the next little while. She also leads the scientific review process for the DPUK data portal and their early career researcher training and mentoring program. And for anyone who wants to go and do a data fun, they have a couple of those, one early in the calendar year and one towards the end of the calendar year that you can see on their website and apply to join. So Sarah is currently visiting Australia. She didn't quite make it to Sydney because she's actually staying somewhere quite remote in outback Western Australia, but she's almost in the same time zone, so that's excellent. And we feel very, very fortunate to have this opportunity to hear from Sarah today on the topic of data curation for multi-modal cohort datasets, the DPUK data model. And just before I hand over to Sarah, I want to just point out that we are recording the session today. Thank you, Sarah. Over to you.
Okay, thank you very much, Vivica, for that introduction. And it's really nice to be here. I would have really preferred to be visiting Sydney, but I think we worked out it would take me an entire day to get there and an entire day to get back. So here I am, a visit another day. So I'm going to just share my screen again. Let's hope that that works. Do it on. Okay, can you see?
Looks good. Yep.
Yeah. Okay. Right. So this session, so the earlier session, for those of you who were not there, I was speaking about DPUK and just giving an overview of the DPUK data portal and some of the science that we undertake in the data portal. And I know that that has been recorded. So I'm sure you'll be able to catch up on that if anything that I say doesn't really make sense. So yes, I've been working for DPUK actually for 8 years, and it's a data repository for over 65 cohorts across 3.5 million individuals, and those cohorts are incredibly diverse. Some of them are birth cohorts, some are healthy population cohorts, some have been collected for cardiovascular disease, Huntington's disease, or even Parkinson's disease, but they're all being repurposed for dementia research. They also come in various formats, both data format wise, but also in the types of measures that they have included. So for example, that you might have depression across most of the cohorts, but that can be measured in many, many different ways, from someone being asked, did you feel depressed in the last 2 weeks, to have you ever visited your physician for depression, to a full scale such as the CESD or the GHQ. And very early on, we realized that there was a problem for us trying to visualize our data on the website. And so John Gallagher at the time came into my office and said, Sarah, you've got to come up with a solution that we can actually bring all the data and show people what we've got. He says, go around the country and find out what everyone's doing. So I did. And I held a couple of workshops and I spoke to existing cohorts about what they were doing. We looked into the OMOP model. We looked into this SNOMED. We had colleagues in Europe using their own model. And we came to the conclusion that none of these models really suited heterogeneous multimodal data sets. And there was only one solution, and that was to create our own ontology or data model. So we call our data model CSERV because it suits cohort survey data initially. And this is the model that we're working with at the moment. It has evolved over the last 5 or 6 years, but we have basically 18 top level categories. And these categories, many people say, well, how did you even come up with them? And we really came up with them after years of just understanding cohort data and how we can come up with top level categories which would fairly represent our data. We had an 18 category, then we went to 22, then we came back down to 18. But we're fairly happy that the categories that we've got now represent most, if not all, cohort data sets. So we've administration will be, for example, anything administrative that is specific to that cohort. So each cohort have done things in various ways, either by touch screen, in person, and that administration category will cover all of that, all the way down to socio-demographic, early life environment, medical history. We have referenced that to the ICD-11 coding. And we did that a few years ago, knowing that the world would be moving from ICD-10 to 11. So we decided to future-proof our model, and medical history is divided into ICD-11. Family disease history, likewise, is ICD-11 codings so that it can easily be mapped onto, for example, the OMOP model. Psychological status, cognitive status, and all the way down to we've got our device-derived phenotypes. Now, importantly, we've got imaging here, and we also have omics. Now, we have not tried to reinvent the wheel for these modalities of data. We admit that for genomics data, we already have SNPs. We already have a well-established standard format of data, and we're not going to try and change that because it serves the purpose. So what we do in our model is we use our model to visualize availability within the multimodal data set categories. The same with imaging. BIDS format is a perfectly good standardized format for imaging data, and we're not going to try and reinvent the wheel for those data as well. So this is a top level of our CSERV ontology, and here really shows the need for standardization. On the left, you can see, I think it's around 13 or 14 of our cohorts and what they call ID. The simple, you'd think that the word ID, that's the first word that comes to people's minds for ID, but no, these are the different raw ID terms that people call ID across just a handful of our data sets. And so another aim in our data curation is to actually standardize the naming structure of variables so that if a researcher applies for 5, 6, 7 cohorts, they know that ID is ID. They don't have to see all the various cohort differences in that variable. On the right-hand side, we see another example. And this variable is, did you ever have a pain in your chest? And these are 3 cohorts who've named it in the colored boxes. One cohort, it was F_059. One is HEYRA, HEYRA, and one is 6059. Now, researchers know that most of the work is on renaming variables, and we know that researchers are going to rename. But when you have around 5, 6, or 7 cohorts that you're working with, if we've already done the groundwork for you, that if this variable, did you ever have the pain in your chest, is intuitive and named the same, across cohorts, it just simply makes an analyst's job easier. Where else do we use our ontology? We use it to feed into our data visualization tools. We have our cohort matrix, which the data comes into DPUK in all kinds of formats, from SPSS to Stata to CSV to very messy to really well organized. And what we do is my team, I have a fantastic team in Oxford, they will take that raw data and divide it into those top 18 categories so that we can have a matrix that shows yes, no, across the 18 categories, whether a cohort has data of that particular category type. It also feeds the cohort directory, and this time, instead of just the top level, we go down to the next levels of the ontology to show longitudinal and actually scale measures within the next category for data application. So this is a bit more of an explanation about the model. So on the right-hand side, we have medical history, and that is our top level. We have 18 what we call themes. We then have around 160 domains within those themes, and these break down that medical history. And then we have around 700 families within domains. And then, of course, the fourth level is the actual variable name that we standardize, and there are thousands upon thousands. One of our cohorts we've recently done has 20,000 of these. So the example that I mentioned previously, did you ever have a pain in your chest? You can see what we now call it in DPUK, pain chest ever. And I think that's intuitive. I think if you read that, you'd sort of know what that means. And we have a rule for how we do that coding. So for example, subject is in this part, object is in that part. And so we have rules which govern how we name that. And we don't make any subjective decisions. We name that variable according to the label that is given to us. So if a cohort has not used a label, that is when we have problems, because we're then doing a bit of guesswork and we're having to go back to the cohort saying, is this what you really meant? And so that takes a lot of time. As you can imagine, this process is incredibly time consuming, but few years on, we are now getting to a place where we have done nearly all of our DPUK cohorts where we have the data dictionaries and the data available for this piece of work. Now, just to clarify terminology, this process we call standardization. It's not harmonization. What we're doing is we are standardizing the organization and naming structure of data sets. So we refer to it as a standard format. Now, because it's time consuming, we're trying to automate the process, and we know it's a very valuable. We're now getting the same people that told us 5, 6, 7 years ago, you're crazy to even try and undertake this, are now coming to us and saying, please, can you do our data sets? Please, can you work with us as a platform? But it's time consuming, it's complex, it's slow and very resource intensive. So our automation program, which is now well underway, will be automating this using ML models. I'm not even going to try and go into details about those because I've got a clever team doing that. But what this will mean is that we can do this at scale and at pace, and we can develop multimodal pipelines much more efficiently. So that is underway. We're hoping by the end of this year, we'll be able, or early next year, we'll be able to provide a service to others where they can go along to a website, they can plug in what they want, and it can be automated to a respectable level, at least at the metadata side. So I'm going to speak now about harmonization. So as I clarified just now, harmonization for us is a whole different process to standardization. Now, harmonization is really working at the data point side. And some of this work we've been undertaking for one of our tools, the Cohort Explorer. And we started with 30 variables. We've just extended that to 40 variables. And this is where we take the raw data and we harmonize it to a common metric across multiple cohorts. And as you can see, the type of variables we have selected and we've started with are particularly dementia or neurodegenerative disease specific. So for example, pain, sleep, diabetes, these are all risk factors for cognitive decline or dementia, and also family history of dementia and cognition. Now, we can't harmonize all of these in a data visualization way. So for example, for our genomics, as I previously mentioned, we don't want to try and come up with a new harmonization rule. So all we do here in our harmonization is we say whether the cohort has genomic data, yes or no. The same with the biomarker. Do they have CRP? Yes, no. So some of these have a simple yes, no, presence or absence for the purposes of data visualization. So this work we've been rolling out across our cohorts. We've done 15 cohorts to 40 variables, and we're working with the ADDI workbench funded by Gates Ventures to harmonize their cohorts as well. Just trying there. So the benefits and the not so good things about harmonization, we're asked this all the time as well. How can we harmonize big data sets across multiple cohorts? I spoke earlier on in my other presentation about a lot of our cohorts. They already have the power in their own right, and you don't really need to harmonize a cohort with 500,000 with a cohort of 12,000, but it is much more valuable to do validation work. So an analysis run in one cohort, you repeat it on equivalent measures in another cohort. Now, this example is taken from a paper that we've published. We took 4 cohorts, Airwave, ELSA, Generation Scotland, and Memento. And they're all from the UK, except Memento, which is a French cohort. And you can see the difficulty that comes with harmonization. So for example, let's take ethnicity. In the Airwave study, they only have white or other. In ELSA, white or non-white. In Generation Scotland, they define it well, a white, Asian, mixed or other. And then in the French cohort, they are not allowed to record ethnicity. So there is nothing. Now, if we had to try and propose a transformation, people would say, oh, but we should have at least 30 categories of for ethnicity or 20. And you would see immediately that Memento would be knocked out of that, and quite a few of the others wouldn't be able to provide information within those categories. There'd be a lot of blank data. So we've come up with an ethnic categorization of white, black, Asian, mixed or other. We're not saying it's perfect, but for the purposes of data visualization, we feel that it is better than what some of our cohorts offer. The same with education. That is very, very different across cohorts and across countries. And you can see for Airwave, which is a British cohort, they provide information on postgrad, degree, A-level, GCSE, NVQ. But for our French cohort, it is totally different with the baccalaureate and the technical baccalaureate. So we've come up with a more simplified version of harmonizing, such as just postgrad, degree, secondary, and primary. Again, it's not perfect, but with harmonization, sometimes you have to take the minimal possible categories to reflect the most information. Same with cohabiting, the same with housing type. You can see some of our cohorts don't even have information on housing type. So when you go to do harmonization work, you have to be very specific about what is important for your research question and how you might either harmonize or will you go the validation route, in which case the harmonization takes on a different format. So this is an example from our Cohort Explorer, and we have here, I think there is around 12 cohorts, and you can see on the left-hand side, gender. Most cohorts have gender. It's easy to harmonize, male, female, and we can select non-binary or prefer not to reply. It becomes tricky, but each of our cohorts have gender, likewise age. But as we go across to the right, we get diminishing returns. And this is the problem with harmonization. We start to have those more rare variables with less representation across cohorts. So when people go to apply for their cohorts and they expect to have the same variable across each cohort, there has to be a bit of compromise of both harmonization, validation work, and which variables are most important for that particular research question. We have two papers. The one paper is on the CSERV data model, and we also have a paper that we wrote with our colleagues in ADDI, where we actually proposed an exercise of 124 variables for a harmonization, and that is actually underway now, but we're still working on the first 40. So Paminda asked in the previous talk about how do we go about imaging and omics data. Now, obviously, these data types have their own formats, which I referred to already, but we are trying to do our best to make imaging data accessible to non-imaging specialists. And in order to do that, it is first of all to show what we have. We have an imaging matrix where you can have a look at what imaging data types are across our different cohorts. And we give information about, for example, the scanners used and how many participants. But importantly is how do we actually do the processing pipelines? Now, when researchers come and they want to access raw data, we do provide them, for example, with a Linux environment, whether that's Ubuntu, and we provide them on the data portal with the tools for pre-processing and analyzing raw image data. Now, this is, I must give credit to Gaurav Bolero at Oxford University, who has worked tirelessly for the last 2 years on trying to standardize both the QC and the pipeline for MRI, and also resting state functional connectivity. And so the work that our Swansea team are doing, led by Lewis Hotchkiss, is standardizing to a BIDS format as well. So Swansea are now undertaking this work so that when you go to apply for your imaging data, the raw imaging data, you know that a BIDS format will be available. And it will have the appropriate derived data set from the BIDS format in order to run your analyses along with the survey and the clinical data. So we are now investing time into these pre-processing pipelines across both Oxford and Swansea. And in my previous talk, I spoke about a study where we did the pre-processing on the data portal. I'll refer to that in a minute. But there is a paper by Ludovico Graffanti and Gaurav Bolero. If anyone would like to read more about the automated QC process of DPUK structural scans. So this was across 2,400 individuals, 19 cohorts in DPUK do have brain MRI data, and this is raw data. And this is where the development of an automated standard QC process is crucial. And this paper goes through the process. I am not an imager. I have done it many, many years ago, but I think this paper is a good reference point to read more about this. So this is an example of a UK biobank where two pipeline versions have been developed for structural, diffusion, and functional imaging. We have a desktop version which uses pre-installed tools on the desktop, so the tools are all there ready, and also an HPC cluster version which uses a combination of singularity containers. So these are both available for researchers depending at what level they want to come in and whether they are a more competent raw image analyst, or whether they just want to use the derived phenotypes. So a single script is provided which can run everything in one go, with also flexibility. So you're able to amend the script accordingly, according to your individual needs. And as you can see, we have all the software available on the data portal. This is an example, a scientific project which I spoke about in the earlier talk, where one of my researchers converted the DICOM files to nifty images through a Linux environment. She reorientated the images, she cropped the images, she corrected them, and she extracted out her IDPs. And she ran this entire pipeline on the within a Linux environment on the data portal. And this was in 468 cognitively healthy older adults. And she was looking at extreme maternal control. This is more abusive control, and whether it was associated with smaller caudate and putamen volumes and also lower well-being. And she found that as maternal control increased, and so the volumes of the caudate nucleus and putamen decreased, and so did self-perceived well-being decrease as well. So this is really important work because as Delia has written in the lower left hand, that this is a long follow-up. These people were born in 1946. We have over 28 collection points for these individuals right up until more recently, a couple of years ago, and we're able to take this snapshot of image data and find long-lasting signatures of early development in the brain and the impact on later life outcomes. And only by using longitudinal cohorts are projects like this really possible. Moving on to genomics, again, I'm not a genomic specialist. Amar is my genomic bioinformatician, and he is working on standardizing the pipelines exactly the same as we've done for imaging for genomic data. So the idea is that someone who is not a genomic specialist will be able to come in, follow the scripts and processes, and be able to explore our genomic data sets. First of all, to interrogate the genomic data across DPUK, is one way to do this is to look at polygenic risk scores. This is an approach which combines the small additive effects of many loci across the genome. And it is really a powerful tool to look at the risk of developing disease. And you can view the distribution, frequency of risk within the cohort or population. And this is Amar, who has shown an example from one cohort. He shared this with me just this week, actually, how he has pre-processed the raw genomic data to work towards creating these polygenic risk scores. Now, we have a project underway in DPUK where we have requested all the genomic data And this is being run with Professor Valentina Escott-Price at Cardiff University to create polygenic risk scores for dementia in our DPUK cohorts. And these risk scores will be available on request through DPUK. So the genomics pipeline, these are the cohorts that we've started with, which is Brains for Dementia Research, Generation Scotland, EPIC Norfolk, NSHD, Airwave 1, and... Okay. Yeah, I'll carry on talking. And as you can see, the genetic data received from these cohorts were different from neurochip to Axiome to the Neurox to imputed with HRC Affymetrix and the Illumina core exome. The population, the number of SNPs that have been extracted also vary from 400,000 to 11 million. And these are worked out on populations varying from 570 through to 21,000. The phenotypes which are available along with these genomic data, what we did was we've extracted out a standard group of phenotypes to focus on, and that was sociodemographic, cognitive status, mental health, and in some BDR, we had the MMSE, but where we didn't have the MMSE, we have extracted out cognitive status. So analyses of genomic data with different data cohorts is really essential to move forward and understand dementia pathophysiology. And if we can compare these cohorts across each other, using a standard pipeline which protects for standard quality control and merge these genomic outcomes, this can be an incredibly powerful resource for researchers. So how are we doing it? Well, first of all, and this I have to give credit to Amar, we do a quality QC of the genomic data using standard software. The standard software that we're choosing is Plink software. So the script will be done through Plink. Then we'll be doing the imputation and harmonization within the HPC cluster. So DPUK has an HPC cluster for genomic analyses, Minimac3. We will extract out the summary statistics from the GWAS results from a different cohort, and then create the polygenic risk score through the Plink software. And then this will be then available for researchers for further analyses and validation of the results. So I have to emphasize this is a work in progress. It's not as developed as our imaging pipeline. This only began about 8 months ago, but we are confident that by the end of next year, early spring next year, pre-processed genomic data will be available for researchers, as well as the polygenic risk scores, which we're working on for both dementia and cognition. That is in the future pipeline as well. Oh, I've just said this, haven't I? Well, I will read the slide because I forgot it was here. What we're also pushing forward as well at DPUK, and this is in the future and ongoing, it will be to look at different disease types and polygenic risk scores. We're looking at high and low cognition and cognitive change over time. And we're also working on the genomic data of the Great Minds registry. Now, Great Minds is another arm of DPUK. We have over 10,000 participants who we've genotyped, and Amar is now pre-processing the genetic data from the Great Minds registry, and that pre-processed data will be available probably from next year for researchers to apply for. We're also working with the Korean Brain Research Institute to look at how human genetic and biopsychosocial data and mouse models converge to further uncover dementia etiology. And this is a really exciting initiative. And to our knowledge, no one is doing this where they work with both the animal models, wet lab, dry lab, in genomic pipelines. So I'm going to keep talking, and this is just a little bit about the training program, and then I believe we'll have a Q&A. So DPUK, we acknowledge that working with large cohort data, multimodal data and longitudinal data is really a step up for most researchers. So over the last few years, we've developed different training opportunities. The first one we developed was the DPUK Datathons. We used to do these in person. During COVID, we took them online. And it is 5 days online, working with 6 of our cohorts, and there are introductions to different cohorts. You team up with researchers across the world, and you interrogate the data with a scientific theme. I actually run these, I supervise these, and you have 3 months continued data access. So there is no teaching involved. You bring your skills, therefore there is a selection process, and these are very popular. So there you have it, the theme, the different cohorts which have agreed to do this. You team up in groups, and then on the final day, you present what you've developed and where you want to take your research. And I'm still working with researchers who've done these datathons over the last few years, and hopefully producing publications out of the datathons. Then we also launched the academies a couple of years ago, and the academies are more training because we recognize that researchers don't have access to affordable, appropriate training for multi-cohort work. And we really look at building on existing data analysis skills and developing skills to interrogate the resources that DPUK offer. These are free to people in LMICs so that we can really democratize training. It's not expensive. It comes to about £125 for 5 days training. And we really want training to be accessible to all researchers across the world. The academies we have available, the Elementary Academy is aimed at people, not just ECRs, but researchers who have either no experience or the experience is a long time ago. And that we run in the spring. We've just done one now in April. And this we go from the beginning to the end to get people kick-started in working in data analytics. The Advanced Academy, and we do have this advertised on our website. It's in November. And the Advanced Academy is really for longitudinal analytics. We do survival analyses on one day, machine learning on another, structural equation modeling on another, and Cox regression. And then the 5th day, it's all pulled together in groups to consolidate what you've learned across 5 days. Again, it's all aimed at being able to work with multi-cohort data. Next year, we will be running an imaging, a genomics workshop, and also structural equation modeling workshops in Stata. So watch this space for additional training opportunities next year. We're also establishing, together with the International Health Cohorts Consortium, a remote mentoring program. We recognize that it's really difficult to get mentors and also to work in a longitudinal data. So using the DPUK portal as a platform for the project, the aim is that we'll have researchers from across the world that they find the researcher, and we help set them up on the portal with a project. And we at DPUK at the moment is just me. I've got 3 people going through the remote mentoring program at the moment, and that they will run their project on the portal with their mentor, with check-ins from a representative from DPUK. They will also link in those that are part of the program with a mentor. They will link into the training program for free. So this is, I just launched this now in Singapore, and this is now a really exciting progression for researchers to have a mentoring from across the world. And that's me. I'm finished. I'm going to stop sharing so I can see everybody.
Great. Thank you, Sarah. That was just a lot of new information there for me. And I bet then also for other people. If people would like to put their camera on, if they're not too shy, that would be nice. Do we have any questions from the audience? Anyone with any burning questions? Put your hand up, or you can put it in the chat and I can read it out for you.
Joshua, start.
Yes, sure. Go ahead.
Oh, hey, yeah, thanks for the talk, Sarah. I just wanted to ask a little bit about your analysis environment. Is it a like fully fledged trusted research environment? And can you tell us a little bit more about the underlying infrastructure, how you provide HPC resources, and what your processes are for essentially protecting the data? Like, do you have like a airlock mechanism? Do you have a committee or anything?
Okay, that's a lot of questions there,
Yeah,
but
sorry.
it's okay. I'll try and answer them. So the environment enables you to access the data securely. There's no internet connection at all, so you cannot download the data. You submit your project proposal, it gets approved. We do a scientific triage at DPUK, then it goes out to the cohort owners. But we host it on a VMware. And I think my previous talk was recorded. So if you look at that, there'll be a more full explanation of what it looks like. But it looks exactly as if you had the desktop on your PC or laptop. Pre-installed tools from SAS, Jupyter Notebooks, Stata, SPSS, R, Python, it's all pre-installed for you. So you don't just get an R console, which is quite common across some of the platforms. You actually get the raw data, you get the statistical tools, and through a airlock system, file in-out system, you're able to extract out your code, your figures, your metadata for publication and presentation purposes. We have a team of data scientists in Swansea who host our infrastructure, our servers, who scrutinize the output to make sure that you have big enough cell values so that no one is identifiable, and that your code, especially for AI projects, doesn't contain or can't be reconfigured for identification of data. So incredibly secure environments. I struggle to get things out myself. That's how strict the environment is.
Well,
Did
thank
that
you.
answer all
And
your questions, Joshua?
yes, just one more clarification. Is the compute infrastructure, is it based on cloud, or is it like a local, is it based in an institute or a high performance compute center?
Yeah, it's an institution. We use University of Swansea host the SERP environment, which has been going for a very long time. It's to the highest security level, ISO. I'm not even going to guess the number now. I might switch it all around. And so literally, if they run out of space, they buy a new rack. I've seen the room, it's huge. They can get really big. We've on purpose gone that route because DPUK really is 10 years old. And at the time, that was the best route to go. And a lot of people have said, oh, don't you want to use AWS or a cloud environment? That gets too expensive, and that goes down to researchers. And researchers, the whole ethos of DPUK is to democratize data access. So if we can cost that infrastructure and give it free to researchers, that's why we do it. The HPC cluster is on cloud, but it's our own. It's hosted. Swansea deal with that. Please don't dig me for more details because I don't know it. But the HPC cluster is available for the genomic and the imaging data. And I know they use buckets and silos and all kinds of things to make it very efficient for storage and processing.
Cool. Thanks so much, Sarah.
No, that's a pleasure.
Thanks, Sarah. Thanks, Joshua, for the question. Does anyone else have a question? Raminda?
Can I ask you, Sarah, about the genomics data? Because that's very sensitive data, generally speaking. What kind of success have you had in getting people to give you the data, especially from outside the jurisdiction, say outside the UK and outside Europe?
Yes, so outside of Europe, not yet. But within even the UK, out of 23 cohorts that we requested, you saw I only presented on 6, because it was only 6 who said, who could see the value in what we're doing. We've actually published a paper on those 6. I forgot to mention that. And so I think it's, you know, trust builds trust. And as people realize how safe the environment is and what we're trying to do, we like to think we've got protection in place that we can't. A lot of people say to us, oh, can we combine, you know, can we see who is in one cohort that's taken part in different cohorts through and no, you know, we really do prevent that to try and keep that security as high as possible. But as I said, this is a work in progress. And I think that I'll probably give different answers in a few months' time.
Sure, because when I was looking at, say, Enigma, the way Enigma deals with it, they don't ask for data. They actually send out the analytic profile or analytic plan to each of the groups and just get the output results and just put them together. So that maybe in the short term, in the medium term, even that may be the way to go, I guess, for most consortia.
Exactly. And I think that that's, again, I go back to our NIH project. I think that most of the 30 consortium members are working on that premise, that this is a way to start, because what we don't want to do is stop good science because we can't move data. Then I think we've really shot ourselves in the foot.
Yeah, but it does put the burden on the contributing study then to calculate, to run the pipelines, right? And then I guess you got your archival studies where there may not be anyone to run anything.
For genomic data, usually they are responsive. There's somebody. I think for the other data sets, studies that don't have genomic data, they often are filed away and forgotten. Yeah, sometimes. Yeah.
Yes, exactly. We do have a couple of legacy cohorts actually who, you know, they finished, they've shut up shop. And we've got one now coming in from the United States, and their funding's come to an end, and they're going to say to us, well, can you manage it now going forward? And I think that more and more grant bodies are going to insist that a trusted environment is used. And data sharing, I'm hopeful it's going to get better, easier.
Yeah, absolutely.
Great. Any other questions from anyone out there? There's a lot of black squares, so I can't tell if you're putting your hand up, but yeah, Shauna, nice to see you.
Nice to see you as well, with Kay. Sarah, that was fantastic talk. Thank you so much. Just a bit of a passion project of mine. I'm out in the low middle income countries. I was really, really pleased to see what you've done in the democratizing it analysis space and with your academies. Are there, from an education perspective, we've had a lot of learnings about the delivery of that, and I just wondered if you could share some insight into that as a first question, if you've had any sort of learnings from running these courses for high income countries to low income?
Yes, so I think that it takes a lot of work from our side. And the main learning is that we didn't cost this into our initial budget. So I do it for free. And I pull in my team and I tell them they get a week off work if they help me with an academy. And so that's learning number one, is that these initiatives need to be costed and placed at the forefront of grant applications, because I've learned the hard way. So I'm actually working with IHCC on a grant application for training. Literally will give us some money to do this. So that's learning number one. And also to really facilitate and to promote it amongst low middle income countries. Because even though you say it's free, you can still attend, it's almost like, well, I don't really believe you, what's the catch?
And I don't belong here. They don't think they belong there. That's the other thing.
Exactly. And I work very hard on simply promoting it, giving presentations. I went to Singapore. I spoke to the South Africans there, and I speak to the people from India. 4 of my team are from India. And so what I try to do is I try to live it as well. It's, as you can hear, this is a passion project. And we match people. So we don't just let everyone come to these academies and these datathons, and the people with less experience who do feel daunted drop off after 2 days. We actually match them so that they work in groups, they encourage each other, and suddenly they feel, oh, I can do this. And I'm going to be developing a workshop specifically for South Africa. Yeah.
Well, we could make that across Africa if you like. Happy to collaborate.
Very happy to do that.
The second question I have is, they're really very passionate across Africa about the continent, which is where I'm working, the space I'm working in a volunteer space at the moment, about the fact there's no African data sets. Like there's very little contribution to any of our imaging studies as far as African cohorts, but I know there's a lot of interest in dementia in specific countries like Kenya, Nigeria, and they're really looking at those cohorts and they're looking at what sort of studies can be done in those spaces. Are there any key sort of documents that I could give to them to, like, where you can influence what they think about acquiring? Obviously, you've got such beautiful learnings from all these studies you've harmonized. I think it would be really helpful to help inform their processes that they're going to go through as they embark on this journey. And just wondered if you could point me in the direction to anything that you think would be helpful to pass on. Or we just get you to come and speak.
Well, I was about to say, what most people do is I just give a talk. And I think that usually is a springboard to people thinking, well, this is what we need. This is who we put you in contact. But I'm very happy to do that. I do it a lot. And because I'm a scientist and I've got a DPUK hat on. So I'm able to do that, but very happy if you want to email me to see what I can do to help you.
That would be great. Thank you, Sarah.
Did you have a comment about this, Paminda?
I just want to say to Shona,
Africa?
That we have a few cohorts from Africa that are part of Cosmic, and we're trying to get data from them. So based in both East and West, really. Eastern countries, also Nigeria. And we are developing an African hub for Cosmic.
Excellent.
Certainly, but as you know, there are a couple of conferences happening on dementia this year in Africa. There's one happening this month, and then there's another one happening in December. And in Nairobi, I think. And I don't know whether Sarah is going to the one in December, but I know someone from Oxford is probably But certainly UK people are quite involved in that conference. And that's bringing together researchers from Africa on dementia. That may be a time, maybe get Sarah to speak to that, maybe virtually, perhaps.
But those studies don't have imaging, right? I mean, the ones that we've...
No, I don't think they have imaging. I think the Nigerians are doing a fair bit of imaging, but those are not population-based studies. Other studies, also stroke studies.
Okay, great. Thank you. Any other questions before we wrap up so people can get to their 4 p.m. meeting?
No, right.
Thanks. Anyone else? Well, thank you, everybody, for attending. You held the audience really well, Sarah, and we've kind of gone a little bit over time. But yes, we've so much enjoyed the last couple of hours, and be talking to the comms team about putting the recordings on the Chiba website, if that's okay with you. And then, we'll send out a link so people can, direct others to it in case someone wants to catch up on what they missed.
Thank you very much, Sarah. Yeah.
It's
Yeah.
a pleasure.
Thank you so much,
Welcome.
and have a
Thank you.
great and wonderful and safe time the rest few weeks here in Australia.
Thank you. Thank you.
Watch
I
out
will.
for the redbacks.
Bye.